In the previous part of this lecture, we talked about the citric acid cycle. Now we're going to talk about the third and final stage of aerobic respiration, which is called the electron transport chain, and we're going to talk about ATP synthase, which is the protein responsible for ATP production via oxidative phosphorylation. So at the point where we left off, which was having completed discussing glycolysis and then the Krebs cycle, we've accomplished the complete oxidation of glucose to form carbon dioxide. All of the electrons that were originally present on glucose have been stripped off through redox reactions, and they have been used to generate reduced coenzymes, specifically NADH and FADH2. Those reduced coenzymes are the point of connection between glycolysis and the Krebs cycle and the third and final stage, which is the electron transport chain. So the reduced coenzymes, which are not attached to a membrane or attached otherwise, they're soluble. So they're able to diffuse to the location of the electron transport chain, which is found in the inner mitochondrial membrane. And once they do that, they participate in the activities of the electron transport chain, which you shouldn't be surprised to hear are a series of subsequent redox reactions. The electron transport chain is going to use the energy generated by those redox reactions to produce something called the proton motive force, and that is going to provide the energy to run ATP synthase. Let's look at the components of an electron transport chain in a little more detail, or in a little detail. So first, what is an electron transport chain? An electron transport chain is a series of complex proteins and quinones. Quinones are small, organic molecules that are able to act as electron carriers. This series of complex proteins and quinones is embedded in a membrane. There should be a space here. Okay. That membrane in eukaryotic cells is the inner mitochondrial membrane. In prokaryotic cells, that membrane is the cytoplasmic or cell membrane. Anyway, the electron transport chain is able to receive and transfer electrons and at the same time simultaneously pump protons or H+, to one side of the membrane. Electron transport chains ultimately deposit the electrons that they transport onto oxygen. Remember that oxygen is the terminal electron acceptor in the process of aerobic respiration. When that oxygen receives those electrons, it is reduced to form water, H2O. Electron transport chains create the proton motive force. Let's just briefly review what the proton motive force is. A proton motive force means a concentration of protons on one side of a membrane. So if we draw a membrane, and all membranes in cells are, of course, phospholipid bilayers. So they look something like this. And you know a lot about their chemistry and their properties. So we've got a membrane. And membranes, remember, are selectively permeable, and membranes do not allow charged species to passively diffuse across them. A proton motive force is an electrochemical gradient, so a proton motive force, or PMF, is an electrochemical gradient due to the accumulation of protons on one side of a membrane. So a proton motive force looks like this. A lot of H plus on one side of a membrane and relatively few H plus on the other side of the membrane. Okay, so you have a concentration and a charge gradient. This side of the membrane is relatively positive as compared to this side of the membrane, something called potential, which is something that one can harness energetically. That is a proton motive force, and it is really the ultimate, or sort of the ultimate endpoint in aerobic respiration. The goal is to make a proton motive force so that you can use the proton motive force to run ATP synthase. The proton motive force is made by the activities of the electron transport chain. Let's think about what's in an electron transport chain in some more detail. So I said a moment ago that an electron transport chain consisted of a suite of proteins or protein complexes and quinones that were embedded in a membrane. So shown here is the membrane. There's our membrane, there's our phospholipid bilayer. Here, by the way, is our proton motive force. Okay, very positive here, very negative here due to the accumulation of protons on one side of the membrane. And shown in green and orange is the electron transport chain. This is the electron transport chain as seen in human cells, for example, and it consists of 1, 2, 3, 4 series of complexed proteins, a protein called cytochrome C, and several quinones. These are the small organic molecules that can carry electrons. All of those are the electron transport chain. In later classes, we'll talk about the details of the proteins that are involved and get into more specifics about what they do. But for our purposes now, let's try to keep it fairly basic. And let's remember that the components of an electron transport chain do two things. They transport electrons and they pump protons. So the electrons are coming from the previous stages of aerobic respiration, glycolysis and the Krebs cycle. They're arriving in the form of NADH and FADH2. Okay, so they came from the Krebs cycle and they came from glycolysis. When they arrive at the electron transport chain, they are oxidized. So the NADH is oxidized to form NAD+, the FADH2 is oxidized to form FAD. Then they go off and get recycled. To be oxidized means that they lost electrons, so they donated their electrons to the electron transport chain. So electrons are essentially passing through the electron transport chain almost like a game of hot potato, where the electrons go to complex 1, then they go to the quinone, then they go to either another quinone and off to complex 3, then to cytochrome C, and then to complex 4. In the case of the FADH2, complex 2 plays a role, and it transfers the electrons from FADH2 to this quinone. Then they again move to another quinone. They're actually on the same quinone, just diffusing over. Then to complex 3, cytochrome C, and complex 4. And at the end of the day, all of those electrons end up on the terminal electron acceptor, which is oxygen, and they reduce oxygen to form water. This is the chemical reaction shown here for the reduction of oxygen to form a water molecule. So that's one of the two jobs of the electron transport chain, the job of receiving electrons from reduced coenzymes, NADH and FADH2, transferring them through the electron transport chain and to oxygen, resulting in the formation of water. As those electrons are transported, they energize these complexes. Specifically, complex 1 and complex 4 use that energy to pump protons. So these arrows shown here and over here on the right, they're showing us that the complex 1 and 4 are pumping protons to one side of the membrane, and that pumping activity is creating the proton motive force. So just a quick kind of recap to focus in here. the electron transport chain has two jobs. One, it receives electrons from NADH and FADH2 and transfers them to oxygen, which reduces it to water. Okay? And the second thing they do is using the energy of the redox reactions occurring, the electron transport chain pumps protons to one side of the membrane, creating the proton motive force. That's what electron transport chains do. And now, finally, we're about to reap the benefits of aerobic respiration, which is pretty much the world's best way of making ATP. And by that I mean we are finally going to get to oxidative phosphorylation, which is the synthesis of ATP by ATP synthase energized by the proton motive force. After the electron transport chain does its work, you have a proton motive force. You can see it here in the concentration of protons on one side of the membrane relative to the other. This is in a mitochondrion. And so the proton motive force in a mitochondrion is organized so that the protons are accumulated in the intermembrane space of the mitochondrion, and ATP synthase is oriented in the inner mitochondrial membrane, such that ATP production will occur in the mitochondrial matrix. The ATP synthase has multiple parts. It's a protein with quaternary structure. It has a component that acts as a proton channel, allowing the protons to flow through the enzyme, and it has a portion that acts as an enzyme catalyzing this chemical reaction, the synthesis of ATP. So ATP synthase does two things: acts as a proton channel and catalyzes the synthesis of ATP. Because so many protons have been accumulated and the proton motive force is such an incredibly powerful force, the activities of ATP synthase are incredibly productive, meaning that you can make a very large number of ATP at this stage. So this is usually upwards of 20 ATP for each one molecule of glucose that originally entered the process of aerobic respiration. So let's think a little bit about ATP production. Which is what the cell really accomplishes by doing aerobic respiration. During glycolysis, we had 2 ATP, and they were made by substrate level phosphorylation. During the Krebs cycle, we had 1 ATP per pyruvate, which is 2 ATP per glucose. Okay? And that's also made by substrate level phosphorylation. But over here at the electron transport chain and oxidative phosphorylation stage, this is where the rubber really meets the mat. And here we get greater than 20 ATP per glucose. Okay? It could sometimes be greater than 30. And these are all made by oxidative phosphorylation. So it is in creating and harnessing the proton motive force to run ATP synthase that you really get the maximal amount of ATP produced, and that is necessary for organisms such as ourselves, large multicellular critters that need a lot of chemical energy in order to run the organism, to do the work of the organism. And so this is really a pretty fantastic evolutionary accomplishment to be able to use aerobic respiration to take advantage of this highly exergonic process, that of glucose oxidation using oxygen to form CO2 and water. And it's very common on Earth. All or many aerobic organisms do this, not all, but many, and certainly all animals do this. So let's sort of sum this up and think a little bit about the locations where all of this is occurring next. Just to do a recap and to think about where things are occurring, we had glycolysis, and this is from the point of view, this is a eukaryotic cell. Okay, and so this is a mitochondria or mitochondrion, a small organelle within the context of the cell. Glycolysis is happening in the cytoplasm. The pyruvate has to be transported into the mitochondria to continue aerobic respiration. The Krebs cycle is happening in the mitochondrial matrix. And the electron transport chain is located at the inner mitochondrial membrane. So the electron transport chain and ATP synthase are at the inner mitochondrial membrane. We can see that the proton motive force, the protons are in the intermembrane space. And the ATP are being produced in the mitochondrial matrix. That's where all these bits and pieces are happening. If this was a prokaryotic cell, there would be no mitochondria, and so glycolysis in the Krebs cycle would happen in the cytoplasm. The electron transport chain would be embedded in the cellular membrane. The proton motive force would accumulate so that you had protons just outside the cellular membrane, in that space between the cell membrane and the cell wall, and the ATP would be produced in the cytoplasm. So before we conclude the topic of aerobic respiration, let's ask the question, what will happen if there is no oxygen? You need oxygen to serve as the terminal electron acceptor in order to accomplish aerobic respiration. But sometimes in cells, the oxygen runs out. For example, in your deep tissue, when you've been exercising very hard and oxygen transport can't keep up with the demand. If there is no oxygen, then the Krebs cycle and the electron transport chain will not run. Okay? So they only happen if oxygen is present. At least in humans, or in other organisms it could be if another electron acceptor is present. If there's no oxygen present, then what happens is the pyruvate undergoes a biochemical process known as fermentation. Okay? And in fermentation, the output is usually a small organic molecule like lactic acid, which is produced, and that production allows the regeneration of NAD+, which then cycles back into glycolysis allowing it to continue occurring, and the glycolysis is producing ATP by substrate level phosphorylation. So the cell will find a way to continue making ATP by continually running glycolysis. And the way that it will do that is by processing pyruvate differently. Instead of sending pyruvate off to the Krebs cycle, it will actually use pyruvate and it will convert it into a small organic molecule such as lactic acid. That's what would happen in humans. In yeast fermenting beer, in ethanolic fermenting beer, such as the yeast that we use to make beer and wine, the pyruvate is converted, of course, into ethanol. But in either case, you take the pyruvate, you convert it into a small organic molecule. That results in the regeneration or production of NAD+, which then cycles back into glycolysis, allowing glycolysis to continue running. And if glycolysis continues running, then you continue making ATP by substrate level phosphorylation, which gets the cell some energy. And with that, I'm going to conclude this lecture. This is the final part of Lecture 12, which addresses the topic of aerobic respiration. And we will then move on into the topic, we will next move on into the topic of photosynthesis in class. Please be prepared to answer quizzes on any of the 3 lectures, the lecture given in class, which was part 1, or the 2 parts of Lecture 12, part 2, which is A and B, of this online video lecture. Please know the content so that you are prepared to take a quiz on any of the information in this material.